Hi everyone. As in the last class of acid base balance 1, we discussed about the what are acids and bases, the pH regulation and the disturbances in acid base balance and their types as metabolic and respiratory acidosis, metabolic and respiratory alkalosis. So further about the topic we'll study in this class. The objectives are basic principles and estimation of blood gases and pH and estimation of electrolytes. So what are electrolytes? Electrolytes are minerals found in body fluids that carry an electrical charge and are essential to keeping the heart, nerves and muscles functioning properly. So as in it's important to maintain a precise and constant balance of electrolytes. They are cation and anion. So the cation are positively charged electrolyte and anion are negatively charged electrolyte. So the cations and anions will be for homeostasis to exist in each fluid compartment, that there has to be balance between them. They are commonly measured in milliequivalent per liter. So which are they? Under the cations, the sodium, potassium, calcium and magnesium, and the anions has chloride, bicarbonate and phosphorus. So first we'll see the sodium as the most prevalent cation in the extracellular fluid. The total body sodium is about 5000 milliequivalent in an adult. Daily requirement of the sodium is about 100 milliequivalent, and in normal individuals the kidney strives to achieve sodium balance, that is to have sodium excretion equal to the sodium ingestion. So what are the functions of sodium? It maintains balance of extracellular fluid, thereby it controls the movement of the water between fluid compartments and transmission of nerve impulses. So it helps in the transmission of nerve impulses and the neuromuscular and myocardial impulse transmission. So how they are regulated? The regulation of a sodium will be by hormones. The hormones increasing sodium reabsorption are renin and angiotensin 2. So renin, it's released from the juxtaglomerular apparatus of the kidney. Release is stimulated by raised sympathetic tone, falling plasma volume and certain prostaglandins such as PGE2. So here no direct effects promoting sodium retention. It controls the renin angiotensin aldosterone axis. So under angiotensin 2, the levels rise as result of renin release. So in turn it stimulates the release of aldosterone, where it also increases tone in the effect of glomerular arteriole. So where this net effect is to enhance the sodium reabsorption from the tubule. So aldosterone, it's a steroid hormone released from the adrenal cortex. So it acts on the distal tubule and collecting duct to increase the absorption of a sodium and water. And arginine vasopressin or ADH, that is anti-diuretic hormone. So neuron cell bodies in the supraoptic and paraventricular nuclei of the hypothalamus. It's stored in posterior pituitary, and the passive absorption of water from the collecting ducts along with a small degree of sodium reabsorption and concentrating the urine. So it acts in the absorption of water from the collecting tubules. And the biological reference range will be in serum 135 to 145 milliequivalent per liter, and in urine for 24-hour urine sample it will be 40 to 220 milliequivalent per day. So it also used in the calculation of a anion gap and osmolarity of a body. And if serum level rise to 160 or less than 120, it will be a panic value. And the potassium, it's a major intracellular cation the total body potassium content in a normal adult will be 3000 to 4000 milliequivalent. That is 98% of a intracellular and 2% will be in extracellular fluid. So what are the functions of potassium? It regulates the neuromuscular excitability and muscle contraction, and also acts as coenzyme, glycogen and protein synthesis, and correction of acid base imbalance. And the regulation will be, the primary mechanism maintaining this balance is sodium potassium ATP phase pump. So sodium potassium ATPase pump actively transports sodium out of the cell and potassium into the cell. So there will be a, in 3:2 ratio, it transports the sodium and potassium ions. And the renal excretion as major route of excess potassium. And approximate 90% of potassium excretion occurs in the urine and less than 10% excreted through sweat or stool. So the biological reference range will be in serum 3.5 to 5.5 milliequivalent per liter, and in urine 24 hours urine sample has 25 to 125 milliequivalent per day. So it's also used in the calculation of anion gap. And less than 2.0 and more than 7.0, there is a panic value. So next we'll see the calcium, the most abundant mineral in human beings. The total calcium in an average adult is about 1000 gram. 99% will be present in bones and teeth, rest in various tissues and body fluids. The calcium is present in bones mainly in the form of calcium phosphate. So about 50% of this bound to protein, and 5% with the organic ions like citrate, and the remaining 45% will be free ionized calcium. So ionized calcium is a active form of calcium. And the functions, as in the formation of bones and teeth, and excitability and conductivity of the nerves, neuromuscular transmission and excitability and contractility of myocardium, and the coagulation of blood also, and in acts as in action of hormones. So calcium regulating factors will be vitamin D, parathyroid hormone, and phosphorus level. So in hypercalcemia and hypocalcemia, so if the level of calcium increases, it's called as hypercalcemia. Where it decreases is termed as hypocalcemia. So there will be a causes for the hypercalcemia as hyperparathyroidism, hypervitaminosis D, and bone cancer, and multiple myeloma, leukemia, polycythemia, milk alkali syndrome, sarcoidosis. And for the hypocalcemia, the causes are hypothyroidism, rickets, osteomalacia, and chronic renal failure, and nephrotic syndrome. So next we'll come to the chloride. That's the total amount of chloride in an average adult is about 80 gram. Chloride in the form of chloride ions is the chief anion of extracellular compartment. The normal serum chloride level will be 98 to 107 milliequivalent per liter. The chloride content of the cerebrospinal fluid is 120 to 130 milliequivalent per liter, and the interstitial fluid contains about 110 milliequivalent per liter. And the functions of chloride will be in maintenance of osmotic pressure, and also acts in maintenance of pH and the formation of hydrochloric acid in the stomach. And the abnormal serum chlorides levels, as changes in the serum chloride level are parallel to those in serum sodium level. So serum chloride level is raised. It's also called as hyperchloremia. In dehydration, respiratory alkalosis, metabolic acidosis, and adrenocortical hyperactivity, there will be a increase in chloride level. And in hypochloremia, that is serum chloride level is decreased. as in severe vomiting, prolonged gastric suction, respiratory acidosis, metabolic alkalosis, and Addison's disease. So next we'll see the estimation of serum electrolytes by using ISE method, that is ion selective electrode. So the principle of ion selective electrode will be the electrode analyzer has different ion selective electrodes consisting of a thin membrane specific for different ions of interest. As example, as separate will be for sodium and potassium and also chloride, so that undergoes a specific reaction with the corresponding ions as such as sodium, potassium and chloride, so contained in the sample being analyzed. So there will be a different ion selective electrodes for the specific ions, and when electrode dipped into the sample solution, a difference in the sodium ion concentration between the sodium solution inside the electrode and the sample forms a electrochemical potential. So between the electrode and the ion containing in the sample, there forms a electrochemical potential, also electromotive force across the membrane of the active electrode. So this potential measured against a stable reference electrode using a voltmeter or pH meter. So the electrochemical potential formed will be measured against the reference electrode which is present in the instrument, and the sodium ion concentration measured is expressed in terms of millimole per liter or milliequivalent per liter. So earlier the blood samples have been tested for electrolyte concentration by flame photometers. So by flame photometer also we can estimate or measure the electrolytes as sodium, potassium and chloride. So later ion selective electrode has come, and despite its relative simplicity, flame photometer is a tedious procedure with high susceptibility to the background interference. So they prefer more ion selective electrodes than the flame photometers. And here the measurement technology used to estimate sodium, potassium, lithium and chloride values in the whole blood and also in the serum, plasma and urine. So what are the advantages of ion selective electrodes? So they are relatively inexpensive and simple to use and no background interference, and they are unaffected by sample color and turbidity. So also can measure both positive and negative ions, and non-destructive, no consumption of analyte, and short response time. In seconds or minutes we can get the result. So this is the picturization of an instrument, ion selective electrode. Here we can see the ion selective electrode which is present in the sample as specific for specific ions, and the reference electrodes. So the main types of ion selective membranes used in ion selective electrodes are glass membrane, crystalline membrane and ion exchange resin. So in glass membrane it's made from an ion exchange type of glass, that is silica of chalcogenide. They are selectively for several single charged cations as H plus, sodium and Ag plus. Example, pH glass electrode. So crystalline membrane is made from mono or polycrystallites of a single substance. So they have good selectivity. And ion exchange resins is based on special organic polymer membranes. An example as potassium selective electrode. So before running patient samples from in the ion selective electrodes, the quality control should be run to ensure the analyzer as in the laboratory procedure. And to run a sample, we need to do all the procedure as given in the SOP, and once sufficient sample has been aspirated, the sample probe will be automatically retract and sample will be pulled into the electrode. So later, after analyzing is completed, the instrument will give the or display the print results. So as in serum and urine, the levels of a sodium, potassium and chloride are given here. So the next, blood gas analysis using ABG analyzer. So the estimation of blood gases and pH by ABG analyzer. So ABG, that is arterial blood gases analysis, is an essential part of diagnosis and managing a patient's oxygenation status and acid base balance. So mainly the disorders of acid base balance can create complications in many diseases states, and occasionally the abnormality may be so severe so has to become a life threatening risk factor. So it's used to test effectiveness of ventilation. And the indications to assess the ventilatory status and oxygenation and acid base status, and to assess the response to an intervention, and to regulate the electrolyte therapy for the patients, and sudden or unexplained and abnormal breath sounds, and also diffuse infiltrates in chest x-ray. So the components of arterial blood gases we can measure in a ABG analyzer will be pH in case of acid or base. To check the, we can check the pH and partial carbon dioxide as and partial oxygen and bicarbonate level, and also saturated oxygen and base excess. So for the sample collection, the choice of site will be radial artery, femoral and brachial. And the procedure of sample collection as a proper blood sample for ABG analysis consisting of 2 to 3 ml arterial specimen collected anaerobically from a peripheral artery, and the ideal glass syringes should be used, and also no air bubble should introduced during sampling. And if repeated analysis is required, use a different site for the sample collection. So this is the procedure, the holding of syringe and needle, and the advance the needle till a blood flashback appears, and also withdraw the needle and syringe and immediately close the recap, and transport the sample immediately to the laboratory, has to be analyzed within the 10 minutes. So this is the part of blood gas analyzer which has amplification unit, has separately for partial carbon dioxide, partial oxygen and pH and reference electrodes, and sodium and chloride. So there, and standard or calibrators etc., and also display keyboard and leads to the print area. And the procedure for this ABG analysis, and the sample has to be mixed with the heparin to avoid the clotting, and if sample is clotted we should avoid the test, and check the machine is ready for measurement of O2, CO2 and pH should be active. So this is the instrument and the ABG report from the instrument as acid base status and the normal value. So this is the procedure as blood gas analysis includes arterial blood with heparin, anticoagulated blood and heparinized blood, so required for the further blood gas analysis. So here we can see the normal blood gas values as analyte comes under pH, PO2, PCO2 and bicarbonate, and also saturated oxygen. So how it will be present in arterial, venous and capillary, those values are listed as the normal pH will be 7.35 to 7.45 in arterial, and PO2 will be 75 to 100 and PCO2 35 to 45, and the bicarbonate level as 22 to 26 milliequivalent per liter, and the base X is +2 to -2, and the saturated oxygen percentage will be 95 to 100. So the interpretation of ABG results, to check from the pH we can identify the whether the patient is acidosis or alkalosis, and the uh, respiratory or metabolic acidosis or alkalosis, and the stages of compensation also we can check, and the simple and mixed disorder, whether it is a simple or mixed disorders, and the oxygenation status of a patient. So this is the instrument of blood gas analyzer, and the normal values for arterial blood gases. So these includes with a complete view of acid base balance. Thank you.